In the second part of the lab, you're going to be looking at a different enzyme and a different reaction. So if you've ever eaten popcorn, you've probably had the sensation that when you put a piece of popcorn in your mouth and start chewing it, it seems to get sweeter and sweeter the more you chew. Well, that's because popcorn and many other plants contain a material called starch, and this is a quick little drawing of starch. Starch is a bunch of glucose molecules held together by alpha-1,4 linkages. So it's this long train of glucose. So what's happening inside your mouth is that these starch molecules are being broken down to simple sugars, maybe just like a glucose molecule, or possibly two glucose molecules held together by a bond. But at any rate, you're breaking down the starch into these simple sugars, and this is what gives you that perception of sweetness. So these are simple sugars. So your challenge here is to figure out whether or not there is an enzyme in your mouth that causes this to happen. In other words, does the conversion of starch, the product, the reactant, to simple sugars, the products, require the action of an enzyme? One thing that you'll need for this activity is distilled water, and there's a carboy of distilled water located at each sink in the lab. Another important thing about this is that anything that has been exposed to saliva needs to be thrown away in the biohazard waste, this red bag which is in the back of the lab. These little cups, you're going to have some reagents we'll talk about, and of course you'll have fresh popcorn. Now, one thing you're going to want to do is chew up the popcorn, mixing it with saliva, and then spit it out into one of these cups. But for example, you might also think that possibly that mechanical act of chewing, maybe that by itself, without the saliva, is enough to break down those starches. So one way you could do that is take a piece of popcorn, you can mix it with some water, and then you can use one of these glass stir bars to mechanically break it up, to imitate what chewing would be like. Okay. So that's one example of the sort of test that you can do. So you're going to have 5 cups. Each group gets just 5 total cups to work with. And then there's also these 2 reagents. There's iodine and ClinTest tablets. One of these is a test for starch. One of these is a test for simple sugars. And I'll let you and your group figure out which is which. One thing important about the ClinTest tablets is that they contain sodium hydroxide. That actually helps them dissolve better, but it also means that if you sat there and held one in your hand for a long time, it would start to melt and you might get a little burn on your hand. So we encourage people to handle them using the forceps here. And when you're ready to use one of them, you can put them in your cup. They need to have a little bit of liquid with them. That could be water or saliva or anything else. And you'll see that they start to fizz up, and it's actually kind of helpful to use one of these little toothpicks to mix it up and make sure that it gets completely dissolved.